The human brain is an immensely complicated organ, so I've set some pretty modest goals for this presentation. We're going to talk a little bit about embryological development and then take a quick look at major brain regions and their basic functions. So as far as embryological development, here's a few important things to know. First of all, the brain and the spinal cord both have ectoderm origins. So recall there are three primary germ layers in the embryo: ectoderm, endoderm, mesoderm, and brain comes from ectoderm. Very early in development, on the dorsal side of the embryo, two long folds of cells form. They're called the neural folds, and those folds rise up and fuse into a tube that's called the neural tube. And that neural tube gives rise to the central nervous system, both the brain and the spinal cord. So our figure here is based on a 21-day-old embryo. So this would be 3 weeks past the point of fertilization. And here we're looking down on the embryo from above, a superior view, and we see the neural folds and how they've fused in the center to form a tube, and that fusion will continue both anteriorly and posteriorly to form a tubular structure with a fluid-filled interior space. Our embryonic development mirrors that of other backboned animals. This is a generic illustration showing a vertebrate embryo in cross-section, any backboned animal. We see at 12 o'clock in that figure the neural tube. So once that tube forms, it drops down below the ectoderm. This figure's color-coded, right? So everything in blue is ectodermal. The red stuff is mesoderm-derived tissue. So a somite is a block of mesoderm that will differentiate into muscle and bone. The notochord is sort of a cartilage-like rod, which is a precursor to our vertebrae. The soma of the vertebra will replace the neural cord and grow up around the neural tube to protect it. So at the head end of the embryo, our neural tube swells, it expands, and it initially differentiates into 3 sections, a set of 3 swellings. We call these the forebrain, the midbrain, and the hindbrain. And we will see that all of our brain structures trace back to one of these 3 swellings. The tube posterior to the hindbrain will become the spinal cord. So here we're tracing the development of the brain through 3 stages: early embryo, late embryo, and then the fully formed mature brain. So in our early embryo, the forebrain is called the prosencephalon, the midbrain is the mesencephalon, and the hindbrain is called the rhombencephalon. Throughout this figure, the features are all color-coded. So we see that as the embryo develops, the forebrain, or the prosencephalon, continues to expand and differentiate. The very tip of it forms a set of structures that are called the telencephalon. The eyes are outgrowths of the forebrain, and the posteriormost part of the forebrain becomes a swelling called the diencephalon. The midbrain or mesencephalon retains its name. The hindbrain, or the rhombencephalon, expands to form two regions called the meton and myelencephalon. So our three-part brain basically becomes a five-part brain. Then as the embryo continues to develop, each of those five regions gives rise to the specific brain structures we will be talking about and those that are illustrated in Figure C. So here's Table 2 in Chapter 13, where that whole sequence is laid out for us. Early embryo, we have three regions: forebrain, midbrain, hindbrain. Late embryo, we have five regions: telon, dian, meson, meton, and myelencephalon. Then we have a list of adult structures. Telencephalon becomes the cerebrum. Diencephalon becomes things like the thalamus and hypothalamus. Mesencephalon becomes the midbrain. Metencephalon gives rise to pons and cerebellum, and myelencephalon gives rise to the medulla oblongata. So you should know that sequence and understand which brain region becomes which adult structure. Now, our original neural tube was hollow and filled with fluid. And as the brain expands and develops, those hollow interior spaces also expand. Our central nervous system has fluid-filled cavities within them that are traced back to the lumen of the original tube. So that's what that column of cavities is all about. Within the cerebrum, we have spaces called lateral ventricles. Within the diencephalon, we have the third ventricle. Within the metencephalon region, we have the fourth ventricle, and the third and fourth are connected by a duct called the cerebral aqueduct. And then within the medulla, we have the central canal. And that would be continuous with the central canal of the spinal cord. All right, now we're going to take a quick look at major brain regions, starting with the cerebrum. In the human brain, the cerebrum is by far the largest region. It is derived from the forebrain. That part of our neural tube expands to the greatest extent of all those regions. So more specifically, it's traced back to the telencephalon part of the forebrain. Our cerebrum has a cortex of gray matter and a medulla of white matter. So when we compare this to the spinal cord, the gray matter was on the inside, surrounded by the white matter. The situation is reversed here. Our cerebrum has a cortex, which is Latin for bark, an outer thin layer of gray matter, unmyelinated, and a core, a medulla of white matter, where everything is myelinated. One of the obvious features of the cerebrum are these folds. Each fold is called a gyrus. The plural is gyri, and that greatly increases the amount of surface area of our cerebrum. We can pack more neurons in if the surface is folded. So here is our figure with the five brain regions. The forebrain expands to form telencephalon and diencephalon, and the cerebrum comes from the telencephalon. Here's a superior view A and a lateral view B. So notice how the cerebrum is divided into two hemispheres, and you can see that reflected in the telencephalon figure. The gyri are obvious. The grooves between the gyri are, each one is called a sulcus, so sulci is plural. So by far, our largest brain region is the cerebrum. As the cerebrum grows, it sort of expands and comes to fold over like the top of a mushroom. It folds over the top of all the rest of the brain structures. In part A of this figure, the brain has been cut and you're looking at it from an anterior view. So what stands out nicely here are the differences between the cortex and the medulla. So follow that brownish colored convoluted outer layer, that's the cortex. All the rest is the medulla of the cerebrum, which is white matter. So remember, myelinated fibers conduct impulses more quickly. And we see connecting the two cerebral hemispheres is a whole network of fibers called the corpus callosum, right? So it's a bridge sort of between the right and left cerebral hemispheres. Also notice in the center a sort of Y-shaped space, right? So those are parts of the ventricles of the brain. Our cerebrum is our awake, conscious thinking part of our brain. So among the important functions would include thought and memory and reasoning. This is sort of where our consciousness resides. The cerebrum is divided into lobes, and the names of these lobes correlate roughly with the names of the bones of the skull that protect the cerebrum. One of the ways that the individual functions of cerebral lobes was determined was by studying people with brain injuries and figuring out if a certain lobe is damaged, how does that affect the person's speech or motor functions or thoughts or behavior. This is a pretty famous case study from the 1800s. A British railroad worker named Phineas Gage, who was using about a 4-foot long iron rod to tamp down explosives. And as he was tamping it, the powder actually ignited and exploded, and it drove the iron rod up through his face and through the top of his skull. This is a photo of Gage with the iron rod, and notice his left eye is closed. In the figure in the upper left, we see a scan showing the pathway that rod took. It went under the zygomatic arch, right through his eye, up through the frontal lobe of his brain. Below that, that's actually Phineas Gage's skull, right? So it preserved pretty well. So we know, for example, that his behavior changed dramatically after that. He became more short-tempered and a bit more angry, and we know the frontal lobe has much to do with behavior and mood. This figure shows the cerebrum from the left view, a lateral view. First of all, I want to draw your attention to this deep sulcus right in the middle called the central sulcus. What a good name for it, huh? Everything anterior to that is the frontal lobe, and posterior to that, the stuff mostly in blue, is the parietal lobe. Then there is a temporal lobe that's kind of pink colored and occipital toward the back. We see mapped on this figure some functional regions of the brain. We know, for example, there's a roughly circular area in the frontal lobe called the motor speech area, right? So that is the neighborhood from which impulses arise that contract muscles that create speech. We have, for example, an auditory cortex, right? So we see in the upper part of the temporal lobe, a darker patch there where we would process auditory stimulus. We have a region, for example, right behind the central sulcus, just above the temporal lobe, where taste is processed, right? So not only do the lobes have certain functions, but within lobes there are functional regions as well. Now, of all the gyri evident in this picture, pay particular attention to the gyrus just anterior to the central sulcus and the one just posterior to it. The regions called the primary motor cortex and the primary somatic sensory cortex. That's what we're going to be looking at in the next two slides. This figure is showing us details about the postcentral gyrus that we call the primary somatic sensory cortex. So that is the fold of cerebral cortex where we process sensory input coming from various parts of our body. If you look at the map in the upper right of the figure, this is showing us exactly where on that fold of gray matter do we process sensory inputs from different parts of our body. And notice how much real estate is dedicated to different areas. Lips, teeth, and gum. Man, there's a big chunk of that gyrus dedicated to processing information from your mouth, and your tongue, and your pharynx, and your hands, your fingers, right? Each finger has a separate area. That is basically a reflection of the density of sensory receptors in that part of the body. The left side of this figure relates to the previous lecture about the spinal cord and the ascending tracts. So if we had a free nerve ending somewhere in your hand, for example, and you touch something sufficiently to generate an action potential, that axon would enter through the dorsal root of a spinal nerve, make a synapse in the gray matter of the spinal cord. An axon would find an ascending tract, pass all the way up through the medulla and the pons of the brain, through the midbrain, hit the thalamus. Another synapse would then route an axon up to the appropriate part of the primary somatic sensory cortex, and you can process that and have some conscious awareness of what you just touched. So this is sort of the sister illustration to the previous one. This is the precentral gyrus, this fold of gray matter right in front of the central sulcus. This is the primary motor cortex. So what we're seeing here is a map of that fold of gray matter showing us the area from which an impulse originates that is headed to muscle in different parts of your body. So if you wanted to move a hand, an impulse would begin somewhere up in that figure, about 2 o'clock or so in the figure. An axon traveling down to the thalamus would conduct that impulse. A descending pathway would be tapped to carry that impulse down to the appropriate local neighborhood of the spinal cord. A synapse would be made with the cell body of a motor neuron that would escape through the ventral root and supply stimulus to innervate the appropriate muscle. So you can create sort of a map of your body as projected onto this fold of brain. They call that the homunculus. So a homunculus is this figure that is projected on your brain that correlates with the parts from which impulses originate. That would be the motor homunculus, and the regions to which sensory information is conducted. That would be the sensory homunculus. All right, I think I'm going to make this the last slide in this presentation. I'm trying to keep these videos short enough to upload without too much trouble and download. So the future presentations will look at the rest of the brain regions.